In this episode of Shareable Science Beyond the Blog, let's talk about vaccines, what they are, how we create them, and why it seems like it's going to take a year or more before we have an active one against COVID-19. Welcome to Shareable Science. Science you can share. So let's talk about vaccines. Right off the bat, these are different than treatments. This is not for individuals that are currently infected with the virus that causes COVID-19. This is given to healthy people to try to prevent them from developing the virus. And the goal is to expose our bodies to something that are virus-like, and I'll explain that in a second, in order to train our immune system to recognize, attack, and destroy the virus if we are infected with it at a later date. So here's, in a nutshell, how this works. And we're going to skip a lot of steps. I'm going to create a vaccine that has something that is virus-like. That could be a weakened virus, so it doesn't actually cause major infection. That can be little pieces of the virus, like the spike protein, or in a twist that we're now looking at for the COVID vaccine, that can be a piece of RNA instruction from the virus that actually tells the body how to make one of the viral proteins. So this is incorporated into the vaccine. It's injected. Many of the current ones in clinical trials are muscular, injected into the muscle. And then the body recognizes the weakened virus or the pieces of the virus or the instructions and says, this is unusual, I've not seen this, and mounts a small but significant immune response. Specifically, for the purpose of this conversation, we're going to look at your B cells, one of your immune cells, which after being activated by the vaccine becomes a plasma cell and is producing large amounts of antibody. Antibodies are your friend. They are one of the key ways your immune system very specifically recognizes, detects, and binds to, in this case, the virus, and says, hey, this needs to be destroyed. So the vaccine generates this entire process at a low level, but it's ready. You've got it in the cupboard if you need it. So if someone is later infected with the virus, SARS-CoV-2, the body recognizes and says, oh, I know what this means. I know what to do with it. Here are the antibodies. Let's start producing large amounts of antibodies. It gives the body a jump start on the immune system, gives the immune system a jump start so that you can quickly detect, attack, and destroy the virus before the infection spreads. Now, the process of bringing a virus to approval, it has to go through a clinical trial, just like other medicines that have to be approved. And there are multiple stages in a clinical trial, each with a different area of focus. We start out with preclinical. This is work done often in a research lab, maybe with cell lines or maybe with animal models to say, would this be a viable option for use in humans? Then you move to human testing, which is divided into 3 phases, phase 1, 2, and 3. Each of those has a different focus and requires a different number of individuals for the study. Phase 1 trials are primarily focused on, is this safe? Does this cause some unexpected set of serious side effects? And it begins to say, and is it also doing what I'm hoping it's doing, but its primary endpoint is really finding what a safe dose is. Phase 2 is now looking at narrowing, getting a better sense of the dose, and is it giving me what I want? So in this instance, is it protecting an individual from exposure to the virus that causes COVID-19? And then phase 3 is with thousands of individuals asking that question and looking at all of the maybe even rarer potential side effects before it then gets reviewed and approved by the Food and Drug Administration, and then you've got to ramp up production to make enough to then vaccinate millions of people. This process is a long time frame. You cannot rush through clinical trials, not if you want to create something that is both effective and safe. So when you hear people talk about the creation of a vaccine as being 12 to 18 months, I know that sounds like a really long period of time. It is actually much, much faster than any other vaccine we've brought to market because so many individuals around the world are working as quickly as possible to move this through. it's likely we're going to have a whole host of potential candidates that enter phase 1 that will get narrowed down. They'll drop out of the pipeline because they don't do what we thought they would, or they have horrible side effects. So we've got to start with a large number so we end up with a handful of candidates that actually we can take to production. If you want to track this, you can go to the Milken Institute. Here's the website. They actually have a tracker that not only tracks vaccine status, but actually other therapeutic approaches as well. Drugs, neutralizing antibodies, and vaccines. Currently, they've got 50 potential vaccines at some part of the process. Most of them are in preclinical. 2 are in phase 1 trials. They've just started in the last month in humans. And 1 will start this next month that's a combined phase 1 and phase 2. So it's now beginning to look at efficacy. Phase 2 and phase 3 are usually blinded. So some individuals get the drug, some individuals get a placebo. In this case, they might get an injection of saline, so that you can determine if the if the vaccine is really doing its job, or if people are dealing with the placebo effect where, hey, I think I'm getting something, and so my body is going to respond. All right, we've covered a lot of content really quickly, but this gives you some background into what the process is to make a vaccine, the approach we have to go through to determine safety, and a way for you to track it. If you find this information is useful, please share this with friends, neighbors, coworkers, whoever you think might benefit from this. Thank you for joining us. I look forward to seeing you again next time. Stay safe, everyone.